I was asked to present Hector, which of course I'm very happy to do. It's a bit ridiculous because most of you know him for a longer time and probably better than me, but still. So Hector received his PhD from computer science in Wisconsin in 2008, and then he went for a postdoc in Hopkins in biostatistics, and in 2010 he has joined our department. And he works mainly on the basic challenge that all of us in our field try to work on. There's tremendous amount of data that is produced, and we are flooded by the data, and we are trying to make some sense of it. Okay? So that's the big challenge. Hector's specific contributions on the thematic side have been on developing different ways to integrate this data, mainly focusing on applications in cancer genomics and cancer epigenetics, as I imagine we will see. And on the methodological side, he developed a host of methods involving machine learning, statistics, and data visualization, and also even numerical optimization. So a multitude of different methods, and we are looking forward to hear.
Thank you, Ethan, and thank you everybody for coming. I'd like to give you a little bit more specifics than what Ethan discussed, but in general, a lot of what we do in our group is kind of take a broad approach to certain problems in computational biology, and at an extremely high level. What does my group do? We seek to understand the molecular basis of variation in development and disease. So by molecular basis, I mean measuring molecular mechanisms in DNA, how that's used, how it is encoded, how it's used, how does it function, how it changes through development, and what might break down as disease arises. And we want to do this using high-throughput methods. And what I mean by high-throughput is a large number of measurements per sample. Might be genome-wide. We're trying to measure something every 6 billion, for over 6 billion bases. It could also be high throughput in the sense of number of samples that we're seeing. We have the ability now to measure a lot more across different populations. And also in the variety of measurements that we make for a given sample. It could be multiple tissues, multiple different tissue types, normal tissue versus their cancer tissues, and increasingly even single cell measurements for each sample. So you've heard of this genomic revolution, I assume, a number of times. And this is, for over a decade, we kind of have been attempting to lay down the basic molecular blueprint of these molecular mechanisms by sequencing DNA. So a lot of what we'll discuss today is based on these vast and fast technological advancements in DNA sequencing that allow us to measure a lot over many people. So luckily, when I started my position here at Maryland as an assistant professor, the NHGRI, which is the National Institute for Human Genome Research, put out their perspective on what their strategic plan for the next few years was, which was extremely convenient for me. Because as the field went on this endeavor, just so happens to align very well with what I wanted to do. And the basic conceit of this idea is we want to get from our ability to measure precisely at the molecular level over a number of, or large number of measurements over a large number of people, to making or deriving knowledge that can help us in treatment and can help us understand the mechanisms, the biological mechanisms of what we are seeing. So at the time, they wrote the data generation problem in biology, or high-throughput biology, we seem to have solved. Right? The biggest problem is in interpretation and analysis, display and integration. And they mentioned these challenges, or these areas that can address these challenges in terms of data analysis, integration, visualization, and computational tools and infrastructure that can get us towards interpretation and discovery rather than just data generation. So it's not an accident that the work that my research group does kind of hovers around many of these areas and tries to touch upon a number of things that we need to do to kind of get into this ability to interpret and understand. My group is involved in kind of just trying to make basic molecular discoveries of what molecular mechanisms are changing over development, over disease. We develop methods to kind of understand what those, or to write those discoveries. And something that my group does a lot of is software tool development to make those methods available to our communities so that themselves can pursue those discoveries. So many of my papers always land somewhere within that simplex, some more in certain corners. When lucky, we hit right in the middle and everybody's happy. So what I'd like to do is tell you a story of one piece of work that touches kind of that space of how my work is distributed toward, around discovery, method building, and developing tools that the community can use. And this story will be based around this notion of computational epigenomics. So what is epigenomics? So we can ask, what makes people different, right? Why one person who exhibits certain phenotypical traits, certain hair color, shapes, or why would people that live apparently healthy lives, take great care of themselves, are more susceptible to, say, pancreatic cancers than others that do not, right? And we understand that a lot of that happens through just differences in DNA. Much of human variation is to difference in about 6 million DNA base pairs, right? So 0.1% of the genome. And most of what we've been trying to do in terms of interpretation, in terms of understanding how we can go from discovery to precision medicine or the base pair to bedside idea, has been concentrating on these differences in DNA, just differences in the sequence of DNA in a particular cell. But something else that we may wonder is what makes, say for example, these pictures of this one individual different, right? What makes a neuron different from, say, a muscle cell? How does different parts or how different structures in cell go from a single cell to a full-grown individual? And in this case, for the vast majority of things that we're seeing, there's no difference in DNA. This is not a difference in sequence itself. These are not inherited differences. But these are instead differences in how information stored in DNA is used differently during different stages and in different tissues. And that's, in terms of epigenetics, is what we want to understand. What are the mechanisms that allows different, at different stages and in different cells, use the single information of DNA that's common to all cells in a particular way? And why do we care about this? Well, we think that many of the processes that are involved in this dynamic usage of the single information of DNA from single cell to a grown individual, or differentiating between different cells, might be the same mechanisms that break down in disease, and that's something that we wanted to understand. So a big part of how this comes about is that actual biophysical object of DNA, that molecule, is packed, right? It's a very long molecule that has to be packed inside a very small cell. So certain parts of DNA become inaccessible through that packing, and this packing is what we think is dynamic. That's part of what changes from cell to cell during time and across tissues. And one specific mechanism that's involved in how that packing changes dynamically is DNA methylation, and that is what we're going to be discussing for a while. It's an extremely simple thing. It's just one little molecule that attaches to DNA at specific locations that has certain properties that make it part of how gene expression is regulated in the genome. So for example, a part of the genome where no methylation, where no modification occurs, may be accessible so that transcription or expression of that particular gene may occur. But by methylation of a particular region, may inside a cascade of processes that could then repress expression of this gene. So you can imagine this might be accessible in, say, a neuron, but this might be inaccessible in a muscle cell, and that's part of what makes them different. And of course, it changes during differentiation, how a cell might become a neuron versus how it might become a muscle cell. And it has a nice little property. We know exactly how this is replicated after cell division. This is what retains memory, in a sense, or identity. That's how a neuron remembers to be a neuron, and a muscle cell remembers to be a muscle cell. So, as I said, these great advances in DNA sequencing allow us to probe processes like these, like DNA methylation, at a high throughput rate. So what exactly are we trying to measure? We're trying to measure for every position in the genome. Well, it's about 9 million of them that we care about, because those are the ones where methylation may occur. We want to measure in those positions in the genome if that particular modification occurs at that position or not. And remember, when we're doing this, we're say, for example, taking a piece of a healthy colon. We have a pool of cells, a pool of molecules, and we're asking how many of those molecules have that modification at that position versus those that do not, and get a percentage that says how many of those have that modification, how many of those do not. All right, so as I said, we have great biochemical tricks to do this. Those positions that we care about, we can do this little trick that if there is a modification there, after this treatment that we can do to DNA, that sequence there will be preserved. Any positions where there is no modification, that sequence will change. So that allows us to say, get over a particular genome, a large number of sequencing reads that say, at that position I observe 3, what I think are molecules that are methylated, which have that modification. So another 100% methylation, 100% methylation, but you have positions where you might have certain cells that might have that modification versus those that do not. So what do we have then? We have data that says for every location in the genome that we care about, in the order of 6 million positions, over hundreds of samples, say samples from normal tissues versus cancer tissues, what is the percentage of methylation observed at a given position? So that's the kind of data that we'll be looking at. And later on, I'll also talk about a different set of data. When we now measure expression over genes, those things that are expressed or not, what are active or not, again, over, say, hundreds of samples in the order of 20,000 genes. And later on, I'll also talk about cases where we have both of these data sets for around 6 million locations in the genome, this percentage of things that are methylated, not methylated, for genes in the roughly 20,000 features, those that are expressed and not expressed, some degree, some real number, and some connection between these roughly spatial locations, so modifications in the genome around the area where a particular gene is located. Right, so that's the data we can obtain. What can we do with that? So the first thing we may want to do is try to have a good estimate of those percentages along the genome. At a given position in the genome, what is the percentage of methylation at a given position? Right, so methylation as a chemical modification has certain properties that there is correlation, spatial correlation as you go along the genome. This percentage at this location is highly correlated to the percentages in the locations near it, and much lower correlation to the percentages that are to the locations that are farther out. So we develop methods to try to get robust estimates of these percentages. We use local likelihood smoothing methods, which have a nice feature. You can think of smoothing as a way of saying how much is the estimate at this given position dependent on estimates around it. And for high frequency smoothing, what we say is that the percentage here only depends on a small number of features around it. Whereas in low frequency smoothing, we would say that the percentage here depends on much longer or much bigger number of locations around it. Which has a nice biological interpretation, because high frequency smoothing estimates local methylation structure. It's kind of what happens in these local areas, versus low frequency smoothing estimates long range methylation structure, which is large domains. So armed with this methodology, we can now start to make certain discoveries. So I was lucky enough to be involved in the first study that ever looked at these DNA modifications over the entire genome, comparing colon cancer to colon normal. It's the first time this was probed genome-wide. And what are the kinds of discoveries that we will make? We will make discoveries of the type, and it's probably the most interesting, is these long range or long domains in the genome where methylation for tumors, so this would be 3 colon tumors versus 3 normal samples, is lost. So what are the important features here? Methylation in normal tissues is fairly well conserved for all 3 samples, so there's little variation across the normals. It is at a partial state, it's at roughly 80%. So it's saying that colon tissue, at least epigenetically, as far as DNA methylation is concerned, is a heterogeneous population of cells. Certain cells have that modification, certain cells do not. Right? And when methylation is lost in cancer, there's more variability observed in cancer, and this variability is more than can be explained by just, if you think of this as some binomial process, what you might see by flipping a coin at 50%, or a fair coin versus a biased coin. So even taking that into account, there's hyper-variability in methylation across tumors. But it has a certain nice properties that the boundaries of these domains, where methylation differences between normal and tumor start and where they end, are very well conserved across tumors. So even the location in the genome is very well conserved, but the levels at which these methylation signals are observed are highly variable across cancer. And it also happens to overlap with a lot of genomic features that we understand are important in determining the structure of the genome itself. And one interesting also fact is that within these regions, the genes that live within these regions are what we call tissue specific. They only express in a small number of tissues. So we think are involved in determining, again, identity of these cells. So kind of leading us to this question that is the same or similar mechanisms involved in determining what makes a tissue, a normal tissue, a specific kind of tissue that breaks down and leads to perhaps cancer development. Right? And what else did we observe? We observed that within these regions, if you look at expression of these genes across multiple samples, comparing, say, for example, colon normal to colon tumors, if you look at those genes where variability in expression is largest across the genome, you will find them much more frequently within these regions of epigenetic change. Right? So, again, given this idea that what we're observing is a breakdown of structure that leads to a certain amount of stochasticity in how information in the genome, namely through gene expression, is used. All right? We observed similar results in a subsequent paper across different solid tumor types, breast, pancreas, thyroid, etc. And again, observed a similar increase in hyper-variability within these regions, by just confirming the same result, but in different cancer types. And finally, if we look for genes that are most hyper-variable, Right? When you compare tumor to normal across a large number of cancer types, we also find that these are also genes that are involved in cellular fate tissue specificity, kind of determining how that dynamic changes across the genome occur. So in summary, one of the discoveries that we were able to make with this type of data is that large domains of methylation loss are a stable mark across cancer types. So the locations where involved are stable, but there is hyper-variability in these measurements in these locations. All right? And that hyper-variability in disease seems to be involved, or seems to be in the same regions that are involved in determining how different cells are, the cell identity and the maintenance of that cell identity. All right? So if we think again to this question of what makes these cells different, we think that it's in the same mechanisms that change during development that determine differences between cell types that break down in a stochastic manner in cancer. So what can we do with that? All right, so now that's a basic scientific discovery. All right? Ideally, what we would like is to translate those discoveries into some kind of clinical action. All right? So molecular methods for cancer detection, for prognosis and treatment will be the basis of kind of what we're looking for in individualized medicine. And for example, gene expression profile methods have been the subject of study for decades. But very few of these proposed predictors are ever translated into the clinic. Right? And by far, the biggest culprit of that is the irreproducibility of results in these preliminary studies. I would argue that if what we're seeing seems to be a consistent behavior across cancer types, is a stable mark that is consistent, or it's a stable mark that is associated with hyper-variability of expression, or a certain stochasticity, that we're kind of in a situation that we're not going to win. Right? Why? Because we take an experiment and we measure over 20,000 of these things. Let's say 5,000, 6,000 or more of those are hyper-variable. Just by chance, if I look, thinking of the machine learning case, if I look for a consistent difference between normal and tumor, just by chance, I may find a small number of those features that exhibit a consistent difference in expression. Right? Just by chance, just because I took a lot of samples over hyper-variable things. Now I take another experiment, I have another sample of this over these high variable features. What's the probability that I'm going to find a consistent difference in exactly the same features? It's extremely low. Right? So part of why these profiles have been not successful in the clinic is we're trying to kind of capture consistent differences between samples over measurements that are hypervariable. So we thought, well, how can we then use these ideas to kind of try to do some predictive work, right? So we came up with this very simple method. We called it an anti-profile score. And what we're trying to do is measure sample-specific deviation from normal expression within this consistently hypervariable gene. So looking back at this figure, these are genes that exhibit hypervariability in cancer compared to normal. I've now circled in blue the expression for one particular subject, so one individual. It's not a consistent difference from normal across genes, even if you think of it in terms of ranks. And again, what we observe is just this high stochasticity in gene expression in cancer. Right? So what we came up with is this extremely simple idea, which we'll then try to complicate a little bit after. Let's just define a range of normal expression, what the normal expression in regular, just healthy colon samples would be. Define a region of expression around it, and as a predictor, just count the number of times a particular gene's expression for a given sample falls outside that region. So in this case, every time the circle is outside this region of normal expression, I would count I'll increase the score. And the higher score, more indicative of tumor. So we were going for simplicity, we're looking for reproducibility, right? So simple is better. So feature selection, just use the ratio of standard deviations in cancer versus normal. A range of normal expression, just take normal expression for a given gene, take the median, plus minus 5 times its median absolute deviation. You can think of this as a L1 version of standard deviation, and just count the number of genes in a sample where expression is outside this normal range. And from that, try to derive a predictor for some useful task in cancer. All right, so the first question is how stable is this across experiments? So we grabbed 2 colon cancer experiments, both with normal and tumor samples. We trained this classifier on one study and tested it on a completely independent study. So what you're seeing here is the prediction rate, this is an AUC curve, or an ROC curve. And what you're seeing here is the prediction properties of one experiment having been trained on data from the other experiment. So this is completely independent. This is not cross-validation. All right? Cross-validation in genomics does not work. It just does not work. All right? We want completely independent experiments. And what we're seeing is extremely high classification rate on this very basic idea. And part of the reason why we think that's the case is because even though these 2 experiments are very different in how tumors are expressed because of this hyper-variability, normal expression across these experiments is extremely stable. So if we look at just the upper bound of normal expression across these genes from one experiment to the other one, we see pretty high consistency. So we have stability in normal expression leading to stability in prediction. Right? And then we did something even a little crazier. We obtained a data set across multiple different tumor types, and we said, let's predict normal versus tumor for one cancer type, holding out all data for that cancer type and training it on every other cancer type. So when you see here that adrenal cortex was predicted with high accuracy, this is determining normal versus tumor for adrenal cortical cancer, having been trained on colon, endometrial cancer, kidney, skin, stomach. Right? So again, remember what we found. We found that there's a connection between this hypervariability and tissue specificity. Right? It's those features, that scientific finding of tissue specificity across and its association to hypervariability that allows us to kind of think of, well, what's consistent across cancer types, and can we use that when we do our prediction? So prediction and leave one tissue out experiment is also pretty high. All right, and of course, if you ask any oncologist, distinguishing between normal and tumor is an absolutely useless task, right? They just cut them up and look. Okay, so that's not particularly useful. Well, something that might be a little bit more useful is to try to distinguish how any differences as cancer progresses from a normal tissue to a growth, but benign, to a cancerous growth. And we looked at this anti-profile score, just deviation from normality in that expression, and see very clear differences between normal adenoma, which is a benign colon tissue, and carcinoma, which is full-blown cancer. And we saw that for colon cancer, we saw it also for adrenocortical cancer. And again, that's a useful one, but on the clinic, what we want to really think about is, can we predict having detected cancer for a person and say treated it either by excision or some chemical treatment or both, what's the probability that that patient will relapse after treatment? or die after detection of this. All right, so we hypothesized, well, if this hypervariability score can capture differences along the progression, then it could also perhaps capture differences in how aggressive these tumors are. And that's essentially what we're trying to do. So these are survival curves. The way to read this is at year 2, what is the proportion of people of this group that survive? This is 60% of the people in this group survive up to second year after treatment. Roughly close to 90% of the people in this group survive or live without relapse after 2 years. All right, so what are these scores? These are just the 33% highest scores in terms of anti-profile, so the highest deviation from normal expression. And this is the lowest scores, the closest tumors in terms of our anti-profile score to normal expression. And here, an interesting finding here is that we're actually using exactly the same features to do this for colon, lung, and breast. Again, trying to find some consistent behavior across cancer types just from this very basic predictive strategy. All right, so in summary, we developed a very simple counting scheme that produces robust, stable, and accurate what we like to call anti-profile. We're not looking for what does cancer look like. What we want is a good estimate of what normal looks like and then capture deviation from that. And that's the important aspect of predicting in cancer. And it has nice prediction properties across experiments, which is what we're looking for, and across tissue types, which I think gives us some pretty deep insight into how cancer behaves. And again, it captures increasing hypervariability associated with progression and prognosis. So I said, all right, well, that's fine, but why might this work? All right? And this is where we try to do a little bit deeper study of the properties of this kind of problem and the kind of predictor that we're building. So for those of you familiar with standard machine learning problems, one very classic problem is anomaly detection, right, where you have samples from one particular class, and you're trying to determine if a new sample belongs to that class or not. Right? So, for example, you have samples from or observations from this blue class. You build a model that describes this blue class well, and then you're able to ask the question, does a new sample that I observed belongs to this class or not? So you see the connection with the predictive method that we were doing. By developing or determining those normal ranges of expression, all we were doing was just building a box around this class and asking how many times in which of those, how many of those dimensions does the sample fall outside the box. So by starting to look at anomaly detection, the problem of anomaly detection in this context, I think we can gain some insight. All right. So, and as we're trying to understand the stability of this type of predictor, my favorite tool of choice to try to understand the stability of predictors is a support vector machine. Why? Because we have pretty good theoretical understanding of what happens to a predictor when you remove one sample, analytically. And I find that useful because that's exactly what happens, in a sense, when you change and go to a new experiment. So thinking of this problem as an anomaly detection problem, and I said you can think of cancer normal as an anomaly detection problem, but that's a useless problem. What we really want is to determine differences between low risk and high risk patients, or samples along the benign versus cancerous samples, all right? So what we really want to do is distinguish observations from two anomalous groups. Say, for example, adenoma versus tumor. So how can we incorporate the fact that we are classifying anomalies, so that we have this sample, this group that is stable, behaves in a fairly well behaved manner, and when we're trying to distinguish between these two anomalies, how can we make use of that stable class to help us? So why and when is it worth doing that? So thinking back as to how a support vector machine may work, support vector machines are fairly straightforward beasts. All the trickery is in this one letter, all right? Otherwise, it's just a standard linear predictor where you have a number of terms in this linear model, each one with a coefficient and some bias, and that you add up to get a score from the sign of which can tell you which class these samples go. And as I said, all the trickery is in this function here. So what does this do? This measures, in the standard sense, similarity in a way between samples. So when you have a predictor, a new sample comes in, you ask, I have this data set over which I built my predictor, I compute similarity from this new sample to all the samples that I had, I weigh them according to some model parameters that I've estimated, and then do a prediction. All right, so if we were to use an SVM to classify between, say, adenomas and carcinomas, between benign lesions and cancerous lesions, between low risk and high risk patients, what we would be doing is asking the question, well, how similar is, say, for example, this adenoma to this cancer? And that similarity, by this hyper-variability idea that we've been observing, will not be a particularly robust measure. So we said, all right, well, how can we get around this? And we decided, well, let's build what we call an indirect similarity. When we measure similarity between adenoma and carcinoma, we do it by comparing the similarity of this sample to all the normals, this sample to all the normals, and from that derive some kind of indirect similarity. Why do we want to do this? We believe that this, going from similarity across this class, might provide more stability. Right? And if we do that, it turns out that's a pretty straightforward operation. You can still solve the standard support vector machine problem where you ask how frequently I classify the right thing, and if this function is wrong, how wrong is it? Plus some penalty that depends on this magical kernel function. Right? And what we have is that we still have a kernel function that has this form, but it's built on similarity between anomalies and normals, and similarity between normals and normals. Right? But we can solve it as a standard SVM, etc. But what do we have? We have this nice property that we wanted to understand the stability of these models somehow. And as I said, support vector machines have this nice literature on bounds for errors based on leave one out, where you leave one sample out and ask, how will my predictor change? And bound the number of mistakes on your data set that predictor will make after leaving that one sample. And that uses stability arguments, actual functional stability based on the derivatives of these functions, etc. So we looked at that and found that under certain conditions, if the kernel between anomalies, when you compare adenomas and carcinomas, is larger in some sense, mathematical sense, between this indirect kernel, and the technical term is that in the positive semi-definite cone, this kernel, the direct kernel, is bigger than the indirect kernel. We can show that the leave one out error bound of this indirect, of the predictors that we can build from this indirect similarity, is bounded by the leave one out error of the standard SVM, and that bound is kind of determined by differences in the eigenvalues of this kernel versus that kernel. And we can use a nice machinery from the SVM path algorithm, some mild conditions to show this. All right? So, what do we find? So if we try to do this prediction of high versus low relapse risk in lung cancer, this is again, in this case, is cross-validation. We can show that a single class SVM, which is related to the anti-profile score that we were using, has slightly better accuracy than a regular SVM, but much lower variability across replications of our experiment. And that this indirect similarity also has higher accuracy than both and maintains this low variability, or this increased stability across replications of this experiment. And we use this also in other data sets that are not genomics, so say, for example, traces of heartbeat traces in fetal data. Again, we can see that this anomaly classification procedure provides higher accuracy and more stability than a standard pairwise classifier. So, in summary, what do we find? We find that profiles learned on this hyper-variability show consistent behavior across tissues and across experiments. We can extend this general anti-profile idea to a functional approximation setting, and using sensitivity-based error bounds, we can start characterizing the effect of incorporating normal observations when classifying between anomalies. So this indirect similarity through normal samples improves stability and prediction performance. And looking forward into this line of work, the next challenges I want to address here is, as I've mentioned before, we've looked at hypervariability across samples, across tumors, across different people. But as I mentioned early on, each of those samples are themselves heterogeneous if you start looking at the single cell level. And I want to start understanding the connection between this within sample, within tumor heterogeneity, and the type of heterogeneity that we're observing across samples. And one way that we're starting to approach this is just using standard, or not using one, extending general frameworks for this type of pattern reconstruction problem from sequencing data, and in this particular case using network flow-based algorithms to estimate the proportion of specific cell-specific methylation patterns in a heterogeneous cell population. All right. So moving forward, what do I want to do in this space? I want to move anti-profiles closer to the clinic. This is what our goal was, and we're moving in that direction. And I wanted to understand a little bit deeper this question of anomaly classification. I think in terms of how we deal with cancer in a predictive setting, this is the right framework in which to look at it, right? We're not, I think it's not very efficient to try to define very well behaved or very stable subtypes of cancer. I think that's going to be a challenge unless we get much larger data sets. But this, just understanding that the framework in which we're operating here is that we have access to the normal behavior of these cells, and we want to understand deviation from that to see how they're really working. And complication in the analysis of epigenomics, as I've described it, is that there is a hierarchical organization to the genome. We'd like to develop methods to understand that, and as I mentioned, better understand this connection between intratumor heterogeneity and consistent hypervariability. So, in terms of computational epigenomics, how do we navigated this space so far? In terms of discoveries, we've discussed this notion of epigenetic differences, how that is related to hypervariability. We've discussed methods both in analyzing the epigenetic data, but also understanding this anomaly classification as a setting to understand predictor stability. But what about the tools aspect? So all of the work that I've shown so far is based on tools that we've made publicly available to other scientists and are in use as we speak. And the tool of choice that we've been using is our bioconductors, this computational statistical analysis platform. We develop and apply methods for analysis in this platform. And for this next little bit of what I'll talk about, the most important thing is our collaborators do analysis with us, with these methods, in this platform, right? I think we're getting close to a generation of biologists who actually can do a lot of what we want them to do. Right? So many of these experiments and studies that we were doing are based on collaborative and exploratory analysis. So thinking about the computational epigenomics problem, we did a lot of data transformation in doing the smoothing questions, applying the smoothing methods, detecting hyper-variability, etc. And to be honest, as we develop these methods, we constantly iterate over how we parameterize these methods and how we use what we know about the genome, coming from other data sets that are publicly available, to really try to understand how well these methods are working. But our collaborators also wanted to do kind of just exploratory understanding of what it is that we're giving them by just searching through the genome, looking for gene names. So, their favorite. Every biologist has a favorite gene. All right, and we wanted to use just a vast amount of data that's out there to understand what we're seeing. So I think all of that work that I described is an instance of what I like to think of, for lack of a better term, genomic data science. All right, we have an unprecedented ability to measure, and lots of publicly available data to contextualize it. And if you think of this, the way, this alternative way in which we do data analysis, where we look at transformations of data, we model, we posit and test and fit models of this data, and then look at visualize the results of these models to understand how well they're behaving. An important part that we need to add in computational biology is this idea of just integrating and contextualizing all our findings based on the vast amount of data that is available. So in the absence of things that make my life easier, we build something, right? That's what we do in computer science, I guess. So this is a tool that we call EpiViz. It's an integrative in the sense that you can integrate large number of federated data sources or distributed data sources. It's a system to implement and facilitate visual and computational exploratory analysis of genomic data. It's browser-based, it's interactive, easy integration of data, helps in reproducible dissemination, and it's based on this idea of having a direct connection between a visualization system and a computational system at the same time. As I said, it's browser-based. If you go to the Nature Methods paper that describes our tool, there's going to be a figure that has a link just like that one. So if you go to the Nature Methods paper, look at our figure and click on that, you'll get exactly what you see in that paper in your web browser or in your slides. So it's embedded now in my actual slide deck. So when I'm having a conversation with my collaborator, I can now actually do analysis as I present, right? So what do? We're doing cool things like being able to link multiple views of the data set. We have expression that we like to measure for, say, colon cancer normal. Over here we can look at the difference in expression across different tissues, which is exactly what we were looking at all this time. We can understand where in the genome we are. I can show you the first static picture of blocks and differences in methylation between normal and tumor, now in an interactive fashion. I can do searches and I can do everything that a biologist wants to do, while at the same time being able to manipulate and transform the data that I care about or fine-tune the methods that we're developing. I think something that we don't really discuss very much in data analysis is the role that creativity plays in just doing effective data analysis. And I think we can go a long way in trying to build tools and software systems that support creative exploratory analysis of these large genome-wide data sets. Right? And how do we, for example, allow that to happen? So as a methods builder, as a data analyst, I might want to explore my data based on statistical guidance. All right, so I calculate a statistic of interest. So for example, I can take my tumor methylation base pair data, compute the regions in the genome that has the highest variability, spatial variability, and explore the genome based on that statistic. So for example, I can create a slideshow that says, go from the regions with the highest variability in the genome and just show them, or show your collaborator as you go along. All right, so based on a statistic that we can compute on the data on the fly, organize your exploration based on that statistic. And this is something my collaborators really dig. Right? But in principle, it's based on this idea of using statistical models to guide exploration of these methods. And in terms of the system, it's a dynamically extensible system. It's easy to integrate new data types and add new visualizations. We gave a lot of thought into how data is represented in this space. So for example, if you wanted to explore the NASDAQ genome, you can look at expression of the Apple and the Amazon gene through time using exactly the same system. We just pull data from Yahoo Finance and go from there. You can see how through time, say for example, Amazon decides to split their stocks, etc., etc. Right? So we're building fairly general methods. So one interpretation of big data is many relevant sources of contextual data. So in order to address that, we wanted systems that easily access and integrate contextual data, driven by exploratory analysis of the immediate experimental data that we're using, that we're transforming, that we're modeling, and supporting that iterative process for visual and computational exploration. And we try to do that with, decent visualization concepts in the background. So moving forward, work that we're doing in this space, along with Nicholas Elmquist at HCIL, we're working on collaborative computational analysis methods. And something that a student in my lab is trying to look into is, once we start looking into collaborative analysis, then questions of security and privacy obviously come up. So we're starting to look along with folks at the MC2 on how we may start to address these questions. And in general, just deeper integration of statistically informed visualization and backwards, so visualization-informed statistical analysis. So now we can fill this last little vertex in our simplex. In terms of this computational epigenomics idea, this is how we try to address real need in the field as part of our own findings and examples. So obviously computational epigenomics is not the only space in which I've done this type of analysis. If you were here for Mihai's talk last week, you would notice that we also did a lot of work in metagenomics. We're looking at mixed genomes. And again, both in the discovery side, we carried out a large study looking at novel pathogenic associations for childhood diarrhea in the developing world, developed new methods for association discovery in metagenomic communities, one genome biology, nature methods, and also, along with that, develop tools that are now being used as part of metagenomic analysis by the community. And again, visualization is going to be a big part of that, where we're developing these methods to visualize hierarchically organized features based on the same systems that we were building. So to conclude, another little pearl of wisdom that came out of that strategic plan from the NHGRI as I embarked on my career was the need of, or the importance of education in meeting those challenges. And they asked us to train and support a new generation of investigators who are proficient in 2 or more of biology, informatics, computer science, mathematics, statistics, and engineering. And the courses I've developed here, I think, are moving in this direction. And I do think we have a wonderful population of students that we can really bring into the fold and make some serious impact in this space. So with that, I would like to acknowledge past members of my group. A lot of this work would not have been possible with wonderful graduate students who are now in great places, who are lucky to have them. So my colleagues at CVCV, who have really been quite fantastic, current members of my group, collaborators at JH and Harvard, and funding from NIH and Second Gates Foundation. So with that, I'll take questions.
Thanks a lot for a great talk. Even I understood. Any more questions?
So you had this anti-correlation idea, and you started with methylation, and you showed that the hypomethylation is correlated with expression. So did you ask whether the variability in expression is perhaps a better indicator than variability in methylation, and maybe variability in some other phenotype like metabolic.
So what we observe is that differences in methylation are consistent with hypervariable expression, right? Are associated with hypervariable expression, right? And it is using hypervariable expression that we then did our studies on prediction and prognosis. Yes,
not
methylation.
Correct.
But the features that we use in all of those predictors are based on either being within regions of methylation difference, methylation loss, or is this consistent hypervariability across tissues. I guess so.
Yeah, so you had this these nice graphs where, depending on where you were in the whatever you call the outlier detection thing,
Yes.
You either died sooner or not. So did all of these patients get the same treatment, or is there some like confounding going?
Yeah, that's a very good question. So it's in terms of treatment that was we took care of. I mean that the studies were based on those that received similar, but not the same treatment. There is some confounding, of course, on other features of the samples, right? Their age, obviously, and their status on other molecular cases, right? Do they have a mutation that makes them receptive to estrogen or ER positive or ER negative? So, in a lot of what we try to do, we try to condition as much as possible to have a homogeneous populations, but obviously that's not entirely possible. So something that we're moving towards is just extending a lot of these models with just the ability to condition on these things in the population. And that's part of where also this kind of support vector machine linear modeling may help us. Yes.
Have you investigated sampling bias? So, for example, you showed this anomaly in normal patients versus the colon tissue of normal patients and the colon tissue of patients with colon cancer. What if you looked at their skin tissue? What if you looked at somebody who had Alzheimer's? Your hypothesis is that hypervariability is due to cell identity. Alzheimer's probably isn't related to cell identity. Do you think you'd see something similar, something different?
Yeah, so that's a very good point. We're looking for a part of our feature selection, right? Is hypervariability in tumor relative to normal. So what we're seeing is this very stable expression of these particular genes in that particular tissue, right? So while it may be that there's certain heterogeneity in the normal population, at least for the features that we were looking at, right? That doesn't seem to be the case. And also, something that's important is that these differences in methylation are kind of consistent with silencing of a lot of these tissue-specific genes. We want them off, essentially, in a colon tissue. And it's really there that then becoming accessible. So genes that should have been turned off in terms of how they're used along the developmental and the differentiation path becoming active again, kind of making a step back and allowing proliferation to occur. So while there might be heterogeneity in the normal tissue, in the normal samples, genome-wide, the features that were selected based on these very precise epigenetic differences alleviate some of that.
All right, one more really quick question. If you had the same patient and you took samples from all of their different tissues, do you think comparing one tissue versus the rest would show those same identity differences?
For some of these genes, but not all. Right? Because not all genes are involved in determining all differences in tissues. Right? So there might be a subset of tissues for which these genes are specific. Yeah. So, I mean, obviously part of moving things into the clinic is really what role does sampling normal tissue prospectively, right? I mean, if you have a bank of normal expression for an individual and see how that changes over time, how would that help you as well?
But the other part is whether the normal tissue from that patient is a better kind of control.
Yeah, exactly. That's what I'm saying.
Yeah,
Any other? Michael?
Yeah, it seems to me that hypermethylation is consistent with other hallmarks of cancer, such as increased transposable element activity. And because, one theory about transposable element regulation is that they're in methylated regions, or methylation inhibits their expression and stuff. And then also, there's some recent work showing that a large part of the recombination, genomic recombination or rearrangement that we see in cancers is probably transposon mediated, because they provide more sites for recombination and things. And so you can see all this sort of multiple effects of hypomethylation that we see with cancers that really might have, fundamentally the same sort of origin, if you will.
Yeah,
so that
so-
pushes the question down to what causes the hypomethylation, right?
Yeah, of course. I don't think we have a universally specific answer to all of that. Well, one interesting finding in that Nature Genetics paper is that we looked at childhood kidney cancer, which is Wilms cancer, and it's particularly well known because of its genomic stability. Right? So there's very little, the classic hallmarks on structural differences and repeats or anything like that. And the same type of changes that we see in methylation, the same variability that we see in expression, the same variability that we see in methylation is also observed there. So at least that's one example in which what we see in terms of methylation changes happens earlier and in the absence of genomic instability. But that's obviously not universal, right? And something else that we observed is that if we looked at something as simple as a copy number in colon cancer, we also observed that the domains of epigenetic difference are much more conserved than the domains of genomic instability, at least in copy number, in a tumor that occurs later in age, but much more unstable. So I'm not saying it doesn't play a part, but there are at least some indications that some of these changes will be either early or even may occur in the absence of genomic rearrangements or significant genomic rearrangements, but it doesn't.
Well, no, it would seem like that. I mean, if you think about it, again, it seemed like hypermethylation would precede a lot of those other changes because you could explain those other changes just on the basis of hypermethylation.
Yeah, exactly. But of course, there's other, that methylation is not lost out of, right? There's the presence of certain transcription factors, certain enzymes that happen, and when those transcription factors become rogue, in a sense, it's still open for methylation. So I don't think we've really solved the chicken and egg problem, but we might have made the chicken and the egg slightly smaller.
Thank you very much.
Okay.
Thanks so much.